that we definitely noticed scientifically is how the terpene profiles change, between a very organically grown cannabis and between a hydroponically grown cannabis. And that's not necessarily to say that hydroponics is negative, because obviously if you've got something dialed in and you're trying to produce a consistent medical product, then being able to do that time and time again is actually very unlikely once you start introducing multiple species into the system, and doing living soil systems. The beautiful thing of a living soil system, though, is obviously, you taste that stuff, you smell that stuff, and it's just so much more dynamic. And we've shown that even some of the terpenes are only produced if microbials are present in the soil, right? If that microbe's not in that dirt, the bud tastes different, right?
Imagine the plant is covered in this invisible, almost like a fungus, and that fungus is operating, it's feeding the roots, it's allowing the plant to break down calcium to the perfect amount. If you look under a microscope, the calcium could look like a boulder next to this little tiny piece of root. But then those living microbials within the soil, they'll come in and do their job, melt down a piece of that, liquefy it, feed it into the root, and that's where your plant starts growing in a way that is, growers get blamed for a lot, but I think, at least in North America where we're growing, an enormous amount of the plants are contaminated, and they just need to be, initiated and cleaned through tissue culture, which is another, a great method of cleaning these microbials and fungi and viruses out of the plant is not the type of tissue culture you might think, which is, most tissue culture is callus tissue culture, and that gets into GMO and all these injecting cells into cells, but this is micropropagation. It's just meristemic tissue culture. It's where the meristem grows out of the plant. They just cut like a couple of little centimeters of that.
Microcloning almost.
Yeah, microcloning, but what they do is they put it into a medium in a test tube, and then that little tissue culture will grow. And if you think this is the cannabis plant and these are the funguses and the bacteria and the viruses, the plant actually grows faster than these can catch up, and because they're systemic, they're in the plant. And so you cut that 3 or 4 times over the course of 6 months. You cut it, you grow it, you cut it, you grow it, and you initiate that plant into a new vigor that when you give that plant to a grower now, they are not even going to recognize at the end. And it's funny because if you gave that initiated plant to a grower and then gave him your Bob's plant food, he'd think at the end Bob's plant food is a miracle, because they finally removed all of these things that were slowing that plant down.
I got a great example of that, and that's a grower gave us some genetics to back up for him, and then he switched from HPS to LED lights. And within about 8 months, his THC numbers had gone down easily 5%, right? And by regrabbing the genetic from us, and with his THC numbers going down 5%, his extraction efficiency was going down significant, right? So obviously, the importance of your lighting and genetic mutation is very important as well.